Hey everyone, we are looking here at section 16.3. As you can see, the title for today is Applications of Biotechnology. So you can see in this picture, we're looking at again this production of genetically modified, or what we call transgenic organisms. And here we're going to look at not just, how do we create these transgenic organisms and what are the tools that we're going to use, but now we're going to talk about some other applications of these types of procedures and methods in terms of furthering our understanding of genomes and treating diseases and so on and so forth. Okay, so again, as always, get the title first. Our objectives that go along with this section, we have three of those. The first one, explain how genetic engineering can benefit agriculture and industry. So we're going to talk again about a couple of different ways in which, these biotechnological tools that we've been looking at throughout the chapter can benefit us in a couple of different forms. How it can be used to help improve human health, and like I just mentioned, address genetic disorders, and then how we can identify individuals, which really goes back to what we talked about previously, which is this idea of DNA fingerprinting, right? So if we consider the first part here in dealing with agriculture specifically, the idea behind creating these genetically modified crops, one of the things you want to keep in mind, the reason why we're doing this and altering our food supply is so we can improve upon our food supply, right? I mean, producing consistently larger, more appealing, more attractive types of fruits and vegetables make it easier to kind of for everybody to access the amount of food that they need, right? And then growing crops in large farms like this with certain technologies, like this tractor that you see, is all of that kind of simplifies this process even more. And the goal for any kind of farmer at any point, especially here in the 21st century now, would be to create the most product, right? Grow the most crops and the best crops so that they can earn the most profit. Now, farmers in general aren't in this business to earn a lot of profit. They don't generally make tons of money. And so dealing with that, they want to make sure that they kind of set themselves up to be as successful as possible. So the idea behind this, again, genetically modified food will give us better, less expensive foods, more nutritious foods potentially, and even less harmful manufacturing processes, to where we don't have to maybe apply pesticides as often. We don't have to apply herbicides to prevent weed growth, that kind of stuff. None of that has to be used because the plants that we're growing are already resistant to all of those and may be able to modify the environment in a way that promotes easier plant growth, okay, of those crops that we need. So we have genetically modified or transgenic crops, genetically modified or transgenic animals. And as we've said before, the animals themselves are not actually in our food supply yet, but we do use these to get other types of products, okay? So when we think about specifically genetically modified plants, one of those that's mentioned here is golden rice. So golden rice is an example of a type of a GMO. It's a type of rice that's been modified in a way to contain excess vitamin A. And the reason why it was created this way, the reason why we would want to have excess and additional vitamin A, is because golden rice can be grown in places like in Southeast Asia, where rice is often grown. And in those same areas, there is an increased kind of prevalence and issue with infant blindness from a lack of vitamin A. So normally, just regular white rice doesn't contain a lot of vitamins or nutrients, but golden rice has been engineered to kind of overproduce some of those vitamins and nutrients. So the idea here is that if those farmers in Southeast Asia can grow this golden rice in place of the regular white rice, they're going to be adding to their country and their continent's really diet lots of extra vitamins in hopes that that will kind of address the issues with things like infant blindness. Okay, so there are some real kind of applications of this in terms of, improving and remedying disease type issues. Okay. We can also use these, as we've talked about before, genomics and genetic testing. And genomics, by the way, is just the study of different genomes and the comparison of those genomes across species. So we can compare a mouse genome to that of a grasshopper if we wanted to, right? And kind of look at the differences in terms of, where these body structures come from, how development may occur, and so on and so forth. The crime scene investigation stuff, we talked about that a little bit with DNA fingerprinting already. But again, this idea of identifying and using evidence that is really DNA evidence that really tends to be more accurate than anything eyewitness or other types of things like that. And so we can use those again to help convict individuals that may have been present at a crime or exonerate those that were maybe wrongfully convicted. Medical research, again, like trying to identify how diseases form, cancer-causing genes, those types of things, all of that would apply here in terms of these genetic engineering practices. Down here with preventing and treating disease, we've talked about some examples of human proteins already, but if you think back to some of those, like when we talked about human growth hormone as an example, insulin as another example, blood clotting factors that are important for people that are hemophiliacs that have issues with trying to clot blood if they get a cut. All of those human proteins now are produced by transgenic bacteria through this recombinant DNA kind of technology, which again involves restriction enzymes, gel electrophoresis, PCR, and sequencing. And so those human proteins can be removed from bacteria where they're produced in massive quantities and then distributed, purified, and given to humans that are in need, that have these disorders or that require additional forms of these, right? So for humans that are diabetic, insulin is kind of a life-saving drug. They absolutely need to have a constant supply of that. And the ability now for us to produce that through transgenic bacteria is really a big deal because it makes the whole process more cost-effective, more efficient, and more people obviously can afford it and access that medication. Gene therapy here we'll talk about in a second, but that's essentially where we're editing genetic information that's already present in a viable living thing. So like in me or in you, that would be the idea. And it does come with a good number of risks, but CRISPR is something that is kind of contributing to that kind of field of study. And the last one there at the bottom, GM mosquitoes, and this may be something that you've maybe heard of in the past. These are genetically modified mosquitoes that either have the ability to maybe the inability to reproduce. So they're basically creating these genetically modified mosquitoes that are sterile so that they don't have the ability to increase their population numbers, or GM mosquitoes that don't have the ability to harbor infectious parasites, or even potentially GM mosquitoes that are all of one gender or all of one sex so that they don't, again, have the ability to reproduce. Because mosquitoes, we know, are not necessarily the most beneficial part of any living ecosystem. So DNA fingerprinting here was something that, again, we talked about previously, and it involved use of restriction enzymes, use of gel electrophoresis, and use of different DNA samples. And so, again, we can use this to identify individuals. This is one of those applications, again, that's just not simply creation of crops for our diets, right? So one thing to kind of understand that we haven't talked about with this is that when we talk about DNA fingerprinting, we're not relying upon the genes themselves that are what make us humans. The pieces that actually differ for us, the 1 in 1,200-ish bases that differ among any two humans, those differences come in what we call non-coding DNA right here. So non-coding DNA is all the stuff kind of in between our gene sequences. So it's the stuff that really, for a large number of scientists, we don't quite understand what its actual job or role in the cell is. We think it may have a regulatory function over other types of genes, but this non-coding DNA, for all intents and purposes, is never expressed in the form of a protein. It's just kind of sitting there. So if we look at this, for example, you can see they've labeled gene A, gene B, and gene C, and I have all this non-coding stuff kind of stuck in between, right? If I compare that, here would be maybe another individual's chromosome, and you could see my number of repeats here, and those non-coding sequences differs a little bit, at least in terms of size, right? So when we do something like DNA fingerprinting and we chop up DNA information with these restriction enzymes, we're obviously going to get different sizes, right? Different sizes of fragments. And when we run those out on a gel to separate them, we get what we call different banding patterns, right? So in forensics like this, if we have suspect 1 and suspect 2, and then maybe this is DNA sample E collected from some crime scene somewhere, suspect 1 clearly doesn't match the pattern that we're seeing here in E. But suspect 2, on the other hand, definitely appears to match that same pattern. And so that matches what we're looking for in trying to identify and place individuals in particular areas. And remember, as I said before, this is not just for forensics. This can also be used to identify biological parents of a child and so on and so forth. Okay, so this is that whole idea, though, of, hey, I have DNA evidence of something. What am I proving or disproving with that? Okay. All right, and then the last one, this is it, I think, for 16.3 and 16, really. Let me scoot myself back down. So gene therapy was the one I said I would come back to, and here we are at gene therapy. This is something that we're now kind of using in combination with CRISPR to try and remedy issues that we have in terms of genetic disorders that are inherited. So what gene therapy is, this is a technique or a procedure that's designed to try and fix mutated genes. So we're trying to fix genes that normally cause disorders or diseases. And the idea would be we have to find a way in this case. If we're talking about a living, breathing human being, if you're going to undergo gene therapy, that essentially means you're going to have to somehow find a way, depending on what the disorder is, to get the correct gene inserted in place of the mutated gene in every single cell that's impacted. So if we're talking about a disorder like leukemia that's kind of affecting everything, all the white blood cells in the body, this becomes super complicated, right? Because you have them all over the place. If we're talking about a disorder that specifically affects the eyes, like the ability to see something, then potentially you could just do gene therapy in that one location. But if it's something that's kind of affecting every single cell, theoretically, you'd have to impact every cell with this type of therapy. So the goal, again, is to get a normal gene in the place of a dysfunctional gene. And so what that means is you essentially have to take out the dysfunctional gene, remove it, and then put the new one in its place, right? The correct one in its place. CRISPR can help with that, right? Because it has the ability to kind of directly edit genetic information as it sits in our cells right now. But beyond that, the kind of the routine that was used previously was to use viruses. So this is an adenovirus, which is also, this modified version in some of the vaccines for COVID-19, which you've probably heard about. AstraZeneca uses a modified adenovirus to get copies of that COVID-19 viral RNA, in that case, into the cell so that the cell can become acquainted with that and kind of mount an immune system prior to being infected. So these adenoviruses are things that normally can kind of make us sick, and they're really good for that reason at attacking cells. So if you put the corrected copy of a gene in an adenovirus and it infects a cell, then ideally that corrected gene will eventually become integrated into the host genome or into the chromosome. But the catch with this is, unless you're using CRISPR and Cas9, this viral DNA and this gene of interest, the corrected gene, can end up anywhere in any of your 46 chromosomes. And in the past, what this has essentially led to are issues for the patients that are undergoing these gene therapy trials, because if that DNA integrates into part of a chromosome that's responsible for something else, it could lead to other types of more detrimental effects. So some, for example, have developed cancer because it's accidentally kind of knocked out a gene that's in charge of controlling the cell cycle. Some patients have died from gene therapy trials. And so for a long period of time, there was not a lot that could be done about this, again, until CRISPR was kind of recognized and developed. All right. And so gene therapy is likely something you're going to hear a lot more about as you get older, and these procedures and methods become more and more commonly used. Okay. So that is all I think I have for, chapter 16. So again, if you have any questions for me, as always, let me know. Get ready for that quiz coming up, and I'll see you next time. Bye, y'all.